Welcome back to the GMP and Quality Fundamentals series, and this is video 13. If you completed video 12 on root cause analysis tools, welcome back. If this is your first time, you can find the full series starting from lecture 1 on this channel. Today we are entering level 2 of the series, and we are starting with one of the most important mindset shifts in modern GMP. Risk-based thinking and the ICHQ-9 principles. This is not just theory. Regulators expect it. Inspectors ask about it. And once you understand it, it changes the way you make every quality decision. Let's get into it. Here is what we will cover today. Number 1, you will understand what risk-based thinking actually means in practice, not just as a concept. Number 2, you will learn what ICHQ-9 is, where it comes from, and why it matters for every regulated industry. Number 3, we will break down the 3 core components of risk: likelihood, severity, and detectability. Number 4, we will look at 2 of the most widely used risk assessment tools, FMEA and HACCP. And number 5, you will see exactly where risk-based thinking is applied in your daily GMP work. From deviations to supplier management to batch release. So what is risk-based thinking? Here is the simplest way to understand it. Think about a hospital emergency room. When patients arrive, the doctors do not treat them in the order they arrived. They triage. The patient with a heart attack gets seen immediately. The patient with a minor cut waits. The resources go where the risk is highest. Risk-based thinking in GMP is exactly the same idea. In the old approach, every deviation gets the same response. Every supplier gets the same audit. Every process gets the same level of validation effort. Same priority, same resources, regardless of the actual risk. In the risk-based approach, you ask 3 questions. What is the chance this goes wrong? How bad would it be if it does? And would we even detect it before it reaches the patient? Based on those answers, you decide how much effort, time, and resources to apply, and this is not optional anymore. The FDA, EMA, and other major regulatory authorities now expect risk-based thinking to be embedded in your quality system. When an inspector reviews your deviation reports or your CAPA system, they are looking to see whether your response was proportional to the risk. If a minor documentation error received the same level of investigation as a potential contamination event, that itself is a finding. The core question of risk-based thinking is, where is patient safety most at risk, and am I focusing my effort there? Now let us talk about ICHQ-9 specifically. Because when we talk about quality risk management in GMP, ICHQ-9 is the foundation document. ICH stands for the International Council for Harmonization. It is the body that brings together regulatory authorities from Europe, the United States, Japan, and other regions to create unified guidelines for the pharmaceutical industry. ICHQ-9 was published in 2005 and was revised in 2023. It defines how pharmaceutical companies should identify, evaluate, control, and review risks to product quality. It is recognized by the FDA, the EMA, Health Canada, the TGA in Australia, and virtually every major regulatory authority in the world. If you work in any regulated pharmaceutical environment, ICHQ-9 applies to you. It applies to all GMP activities: manufacturing, quality control testing, validation, storage, distribution, and more. And the core principle of ICHQ-9 is this: the effort you apply to managing a risk should be proportional to the level of that risk. A higher risk situation deserves more resources, more investigation, more controls. A lower risk situation does not need to consume the same level of effort. The key output of a proper risk management process is a documented decision. You have assessed the risk, applied controls, and you can justify why the residual risk is acceptable. That documentation is what regulators look for. Before we go into the tools, I want to make sure you are very clear on the 3 components that make up risk. Every risk you encounter in a GMP environment has 3 dimensions, and you must consider all 3 together. The first is likelihood. How probable is it that this failure will actually happen? The second is severity. If the failure does happen, how bad is the impact? Specifically, how harmful could it be to the patient or the product? And the third is detectability. If the failure happens, how likely are we to catch it before it causes harm? Before it reaches the patient? These three together determine your overall risk level. A failure that is highly likely but has very low severity and is easily detected might actually be lower risk than something that is rare but catastrophic and completely invisible until it is too late. This is why you must never assess risk on just one dimension. All three must be considered together. Let me go deeper on each component so you have a real practical understanding, starting with likelihood, also called probability. This is how often the failure is expected to occur. You might rate it on a scale of 1 to 5, or use categories like low, medium, high. For example, if an analyst has been fully trained on a well-written SOP, the likelihood of them making an error is low. If there is no SOP at all and the process relies on individual memory, the likelihood is high. Severity is about impact. In pharmaceutical GMP, severity is almost always anchored to patient safety. A critical severity would be something like a contaminated injectable product that could directly harm or kill a patient. A minor severity would be something like a mislabeled internal document that has no impact outside the building. Critical severities always demand the highest level of response, regardless of how unlikely they seem, and detectability is perhaps the most underestimated of the three. This asks, if the failure is happening right now, would we know? A visual defect on a tablet is easily detected during inspection. A microbial contamination below the limit of detection in a finished product has very low detectability. When detectability is low, the risk automatically increases because you may not find out until it is too late. Keep these three in mind every time you assess any quality event. ICHQ 9 defines a 4-step risk management cycle. I want you to remember this cycle because it applies to every risk situation you will ever face in GMP. Step 1 is risk assessment. This is where you identify the hazard, analyze how likely and severe it is, and evaluate whether the risk is acceptable or requires action. This is the thinking step. Step 2 is risk control. Once you have assessed the risk, you decide what to do about it. You can accept it, eliminate it, or reduce it to an acceptable level. The important thing is that your decision is documented and justified. Step 3 is risk communication. Your risk decisions need to be shared with the right people. Management needs to know about high-risk situations. Regulators may need to be informed in certain circumstances. Transparency is a regulatory expectation. And step 4 is risk review. Risk management does not end after step 2. You must go back and check. Is the control still working? Have conditions changed? Has the risk level changed? Risk review feeds back into the beginning of the cycle. This is why I call it a cycle, not a process. Quality risk management never fully ends. It is continuous. And that is exactly what ICHQ 9 requires. Now let us look at the tools. The first risk assessment tool we will cover is FMEA. Failure Mode and Effects Analysis. FMEA is the most widely used quantitative risk tool in the pharmaceutical industry. It gives you a structured way to list every potential failure in a process, assess each one on all three risk components, and calculate a priority score. Let me show you how it works. In FMEA, you build a table. Each row is a potential failure mode in your process. For each failure mode you record, what is the failure, what is the effect if it happens, then you score it on severity, likelihood, and detectability, each on a scale of 1 to 5. And then you calculate the RPN, the risk priority number. The formula is simple. RPN equals severity multiplied by likelihood multiplied by detectability. Look at the examples on this slide. Raw water used in mobile phase gives an OSH-PLC result. Severity 4, likelihood 2, detectability 3. RPN is 24. Medium priority. A temperature excursion in storage causes product degradation. Severity 5, likelihood 3, detectability 4. RPN is 60. This is your highest priority. It needs immediate action. A calibration expired on a balance gives an incorrect weighing. Severity 5, likelihood 1, detectability 2. RPN is only 10. Despite the high severity, the likelihood is very low and we would detect it quickly. Lower priority. The RPN does not tell you whether a risk is acceptable or not on its own. Your organization sets thresholds. For example, any RPN above 50 requires a CAPA. Any RPN above 100 requires immediate process shutdown. These thresholds are defined in your risk management procedure. FMEA is especially powerful during process design, equipment qualification, and review of recurring deviations. The second tool we will cover is HACCP. Hazard Analysis and Critical Control Points. If you come from a food industry background, you will already know this name well. HACCP originated in food safety and has been adopted into pharmaceutical GMP because it is a very systematic way to identify and control hazards. Where FMEA gives you a score for every failure mode, HACCP focuses specifically on identifying the critical control points in a process. These are the specific steps where a control measure is absolutely essential to prevent or eliminate a hazard. The 6 steps of HACCP are as follows. Step 1. Identify the hazards. What biological, chemical, or physical hazard could harm the product or patient at each stage of the process? Step 2. Identify the critical control points. These are the steps where the hazard must be controlled. Not every step is a CCP. Only the ones where control is critical. Step 3. Set critical limits. For each CCP you define the maximum or minimum acceptable value. Temperature must not exceed 25 degrees. pH must be between 6.5 and 7.2. These are your limits. Step 4. Monitor the CCPs. You establish a system to measure and record the critical parameter at each CCP in real time or at defined intervals. Step 5. Define corrective actions in advance. If monitoring shows a CCP is out of its critical limit, what do you do? This is decided before anything goes wrong, not during the crisis. And step 6. Verify and record everything. Confirm the HACCP plan is working. Document all monitoring data and corrective actions. In a regulated environment, if it is not documented, it did not happen. Now let us look at where risk-based thinking actually shows up in your day-to-day GMP work. Because this is not just an abstract concept. It connects to almost everything you do. In deviation and CAPA management, your risk score tells you which deviations need an immediate CAPA and which ones can be monitored over time. Not every deviation needs a full investigation team. Risk tells you where to focus. In supplier qualification, high-risk suppliers such as active pharmaceutical ingredient manufacturers or primary packaging suppliers receive full on-site audits. Lower-risk suppliers of ancillary materials might only need a questionnaire and certificate review. Risk determines the depth of oversight. In change control, a major process change that could affect product quality needs a full risk assessment and revalidation. A minor administrative change in a document header does not. Risk defines how much validation work is required. In audit planning, audit frequency is risk-based. A supplier with a poor track record or who supplies a critical material gets audited more frequently. A low-risk, well-performing supplier gets a lighter touch. In validation and qualification, you use risk to identify which process parameters are critical and which are not. Your validation effort focuses on the critical parameters. This saves time and resources while keeping the focus on patient safety. And in batch release, QA uses risk to make release decisions when there are borderline results or minor deviations. The risk to the patient guides whether to release, hold, or reject. Risk-based thinking is the thread that connects all of these activities together. Let us bring it all together. 6 things to remember from this video. Number 1. Risk-based thinking means directing your effort and resources to where the risk is highest. It is about proportion, not equal treatment of everything. Number 2. ICHQ 9 is the global standard for quality risk management. It is recognized by FDA, EMA, and all major regulators worldwide. Know it. Apply it. Number 3. Every risk has 3 dimensions: likelihood, severity, and detectability. You must assess all 3 together. Never judge risk on a single dimension. Number 4. FMEA gives each failure mode an RPN score. Higher RPN means higher priority for action. Use defined thresholds to decide when a CAPA is required. Number 5. HACCP identifies the critical control points in your process. These are the steps where control is not optional. They are the steps where failure would cause direct harm. And number 6. Risk management is a cycle, not a one-time event. Assess, control, communicate, review. Then start again. Conditions change. Risks evolve. Your management of them must evolve too. That is video 13 complete. You have now started level 2 of the GMP and Quality Fundamentals series. And you have the foundation of risk-based thinking that will support everything we cover from here forward. Coming up in video 14, we go into method validation versus method verification. What is the difference? When do you validate, and when do you verify? This is one of the most commonly confused topics in analytical chemistry and GMP. We will make it completely clear. If this video helped you, please give it a like. It genuinely supports this channel and helps us reach more quality professionals. Subscribe if you are not already, and drop your questions in the comments. I read every single one and I reply. I will see you in video 14.